Hello, I'm Cameron Hayden, and I want to thank you for joining me for today's presentation. Today, I'll be covering more information on the analysis of bovine embryo morphokinetic activity in relationship to pregnancy and parturition outcomes. As we all know, there are two commercially available methods of embryo transfer for the bovine industry. First, if we focus on the left, there's conventional embryo transfer. This is where a cow is treated with exogenous hormones to undergo the superovulation process. She is inseminated, and the embryos develop within the cow, where they are flushed out approximately 7 days later. On the right is the in vitro produced embryo process, where oocytes are aspirated from the donor cow's ovary, matured, fertilized, and grown within a laboratory setting. Then, in both cases, the embryos are transferred into the recipient cow. Let's look at the worldwide trends for bovine embryo transfer on this chart provided by IETS. Here, we can ultimately see a continued growth of ET over the past 20 years. Let's highlight this crossing point where the number of in vitro produced embryos has surpassed the number of conventional or in vivo derived embryos, approximately near the 2015 mark. This is likely due to a plethora of factors that favor IVP embryo production, like the ability to have multiple oocyte collections with less time between procedures, the ability to utilize multiple sires, sex-ordered or high-value semen after knowing the donor has produced oocytes, and the ability for collection procedures to be done on prepubertal and pregnant donors. Yet, even with this increased use of IVP for embryo production, we still run into problems. First, IVP embryos have been shown to have lower pregnancy outcomes when compared with their conventional counterparts. Second, IVP embryos have shown to have higher presence of pregnancy abnormalities, like higher abortion rates, increased tendency for male-produced offspring, more calves that are born dead on arrival, and even calves born with large offspring syndrome. These embryonic differences are non-identifiable to the human eye, even under considerable microscope magnification. This makes it almost impossible for embryologists and practitioners to identify potential embryonic abnormalities at the time of embryo selection and transfer. Ultimately, these challenges cost producers billions of dollars in annual loss and make it a large risk that others just aren't willing to take. Still, as technology has continued to advance, so have alternative solutions at identifying biological differences that are non-identifiable to the human eye. Over the past several years, time-lapse imaging and deep learning models have assessed in vitro fertilization, blastocyst stage, and quality calculations, among others. Even more recently, real-time video of embryo morphokinetics has allowed researchers to quantify time-specific morphological changes during development. And this can highlight important information regarding embryo growth and health. Thus, the objective of this research was to measure embryo morphokinetic activity in relationship to bovine pregnancy and parturition using graphic image processing techniques. This schematic of data collection process starts on the left. The embryo data in this study were collected from routine field practice. These embryos were made in a commercial IVF laboratory and shipped to transfer locations. Embryos were video recorded for 30 seconds with a View 4K camera attached to a microscope. Then, embryos were transferred into recipients, and pregnancy was determined by transrectal ultrasonography at day 30 and day 60. Parturition records were recorded on recipients that achieved a pregnancy through full term, and then embryo videos were processed using graphic image processing techniques. As we continue with the materials and methods, it's important to remember that a video is just multiple still images that, when stacked on top of each other and played in a quick succession, creates visual movement, just like this series of images depicting a horse running. That leads into the embryo video details for the study, where a 30-second video was recorded of all embryos. Each 30-second video was recorded at 30 frames per second, leading to a total of 900 frames for analysis, which were then applied GIP techniques to determine embryo morphokinetics. On the left, we can see the traditional video input. When applied with the machine learning model and computer analysis, embryos were then identified and circled in red for further evaluation by the computer. After the embryos were identified by the machine learning model, the object detection model layered each frame on top of each other and subtracted the pixel difference from one frame to the next. This image subtraction process quantified the pixel change from one frame to the next frame for the entirety of the video, as we can see here in the last image where white pixels are denoting a change. This allowed the capture of morphokinetic movement of each embryo and the ability to quantify the results. For the study, we have defined full term as embryos that achieved a full term pregnancy and resulted in a live calf at parturition. Abortion as embryos that achieved a confirmed pregnancy at 30 days but resulted in a loss at 60 days pregnancy check. Open as embryos that did not achieve a pregnancy at the 30-day pregnancy check, and DOA as calves that were deemed dead on arrival at parturition. As we move into the results section, it's important to note that since the time of abstract submission, there has been several new samples collected with full-term pregnancy outcomes, and so I will continue to present only these updated results. First is this box and whisker plot of morphokinetic activity of in vitro produced bovine embryos that resulted in abortion or full-term pregnancy. We can see that there is evidence for embryos which resulted in abortion having lower mean activity level than those surviving to full term. In another sample of embryos, we also compared embryo morphokinetic activity of in vitro produced bovine embryos that resulted in abortion, full-term pregnancy, or no pregnancy. Again, we found that there were only significant differences between the aborted and full-term pregnancy embryo morphokinetics, whereby the aborted embryos resulted in the lowest activity levels when videoed immediately prior to embryo transfer. It is important to note that in this data set, all calves were born dead on arrival were male. Therefore, we presented the average embryo morphokinetic activity of male dead on arrival embryos and live birth male calves. However, there was no evidence for a significant difference in embryo activity here. We believe this is likely due to the limited sample size. In continuation of the previous slide, however, we have a histogram of birth outcomes of male offspring. We can see that the majority of the male dead on arrival embryos clustered in the activity level near 0.5. This means that with an increased sample size and the use of powerful machine learning techniques, future work may be able to identify these prior to transfer. Ultimately, a summary of our findings is that machine learning analysis and graphic image processing techniques can be used to identify and compare embryo morphokinetics with various pregnancy outcomes. Also, in vitro produced embryos that resulted in abortion have decreased embryo morphokinetic activity when compared with embryos that resulted in normal pregnancy or those that did not achieve a pregnancy, when these embryos were videoed immediately prior to embryo transfer. Further research is ongoing to perform other graphic image processing and machine learning analysis, ultimately to develop non-invasive means to identify these embryos that do not lead to a full-term healthy-born calf. I'd like to acknowledge the team that helped collect this research at Team mGenesis. And thank you for joining me on today's presentation. I'd be happy to answer any questions if you'd like to contact me at the following email address.